In this lecture, we're going to cover the topic of bioelectrical signaling. And before we get into the mechanisms of how neurons generate bioelectrical signals, we look at the overall structure of nerves. And we start with the overall nerve that we see here. And we see that the internal part of a nerve are not just made up of neurons, but also are embedded with blood vessels, as well as surrounding adipose tissue. The outer layer of a nerve is referred to as the epineurium. And as you can see here, this epineurium functions to bind fascicles together. I will define that here shortly as well. And so the outer epineurium will separate different nerves from each other, as well as providing a connection to the tissue within which that nerve moves through. As we look inside the cross-section of a nerve, we see that there are these organized fascicles. And the fascicles are collections of neurons. And those neurons are organized by the peritoneum. So as we have a connective tissue type of covering around an entire nerve, so do we have a connective tissue covering around a group of these neurons. And what we see here with respect to each individual neuron, whether the neuron is myelinated or not, the neuron is surrounded by an exterior connective tissue-based covering called the endoneurium. And the endoneurium serves to isolate one neuron from the other neurons in this organized manner. And once again, the fascicle, colored here in blue, represents a group of neurons, and the axon here at the very bottom illustrates just that one extension of a single neuron. This particular neuron happens to be myelinated by the Schwann cell, and as a review, the Schwann cell is the glial cell in the peripheral nervous system that is responsible for producing the electrical insulation functioning myelin. Now, when we start talking about bioelectrical energy, the cellular component that is responsible for the generation of that bioelectrical energy is the plasma membrane. And there are 6 basic characteristics of plasma membranes. Plasma membranes forms essentially a closed boundary of the cell, and the cell in this case is the neuron. And the closed boundary is not an absolute boundary. The closed boundary allows selective entry and exit of ions and molecules in and out of that cell. The plasma membrane is composed of 2 layers of largely phospholipid molecules. The membrane is embedded with proteins, and we'll address that here shortly. And so the thickness of a plasma membrane does range, depending upon the type of cell, anywhere between 60 to 100 angstroms in terms of length. And 1 angstrom is equal to 10 to the minus 10 meters. The second basic characteristic building off number 1 is that the plasma membrane is largely composed and based upon the foundation of phospholipid molecules, but are embedded with proteins. And depending upon the type of cell, the ratio of lipids to proteins ranges anywhere from 1 to 4 to 4 to 1. So there is a large variability of how many proteins are embedded in that membrane. For example, the important energy-generating organelle called the mitochondria for plural, the membrane is estimated to be made up of 3/4 protein and about 1/4 of lipid. The plasma membrane of many cells are best estimated to have about a 50-50% composition, but once again, that's just a very general statistic because each cell will have varying composition values. Some membranes also contain carbohydrates if those carbohydrates are chemically linked to proteins, and those specifically are called glycoproteins. The third interesting characteristic about plasma membranes is that plasma membranes are both hydrophilic, which translates to water loving, and hydrophobic, which is water fearing. So the interesting aspect of plasma membranes is that it's depending upon the part, it is both repelled by water and attracted to H2O molecules. And because of this dual characteristic, the membrane spontaneously forms bilayers. And that bilayer is ideal by serving as a barrier to the flow of polar molecules into and out of the cell, and that allows the membrane to be selective as to which molecules and ions can enter and exit. The different proteins embedded in the membrane do have a specificity with respect to function, and we'll address this a little bit later in this video with respect to channels, pumps, energy transducers, and receptors. And so these are essentially 4 different functions depending upon the type of protein embedded in the membrane. Membranes are polarized, or set up the cell to be polarized, which means that there is a separation of electrical charge inside and outside the cell. Typically, the inside of the mammalian cell is electronegative with respect to the outside. And the inside of the cell typically has a resting, what's called potential, which is a measure of voltage or energy of negative 60 millivolts with respect to the outside. And this is based upon the distribution of charged elements inside and outside the cell. Those are proteins and ions, so that the net charge inside a resting cell is more negative with respect to the outside. And mechanically, structurally, membranes are not rigid structures, they're actually fluid structures. And one description of the plasma membrane as a fluid structure is that it's a 2-dimensional solution of oriented proteins and lipids. As a result of the hydrophilic and hydrophobic nature, lipid molecules can diffuse quite easily through the membrane in the plane of the membrane. Lipids and proteins typically do not rotate in the membrane, and we'll illustrate that here with this drawing at the very bottom. And what you see here are these phospholipid molecules in what's called the functional bilayer, where the interior part of the cell membrane is considered to be hydrophobic, and the what's called the polar heads of each phospholipid molecule are the polar component that interacts with aqueous media outside the cell and inside the cell. And in this drawing, what we see here are a number of these structures that embed in between these phospholipid molecules. So this structure here, this drawing represents for example, an ion channel. And this ion channel, depending upon the type of channel it is, can allow ions to go out of the cell or into the cell. Now, when the cell begins the process of synthesizing this new protein, that protein can move into the membrane and can actually move back out of the membrane. Because of the nature of these phospholipid molecules, proteins can migrate through the membrane, not just into and out of the membrane, but laterally along the plane of the membrane. But the rotation aspect, typically these proteins are not able to rotate or spin inside the membrane. So continuing with the fluid mosaic model, this is a general model that describes membranes not being rigid. So the phospholipid interior hydrophobic nature of the chemical bonds allows those bonds to form and break and form and break repeatedly, providing a fluid nature to the membrane. And some of the key features of the membrane proteins is that the proteins are involved with transport or attachment of other molecules or ions into and out of the cell. The proteins are important in transducing exterior signals or exterior molecules to the interior of the cell. Now, that's not to mean that the protein will move the molecule into the cell. That's the transport function. A protein will bind a molecule outside the cell, and that binding will initiate a series of internal changes inside the cell. One example is that epinephrine, which is an adrenaline type of hormone, will activate enzymes inside the cell to increase the rate of cellular metabolism. The proteins tend to be larger than the lipids. An individual protein tends to be larger than a phospholipid. As a result, those proteins tend to move relatively slow through the membrane, which is predicted in part because of its size. And the membrane does have immobile proteins. So although proteins can move through the membrane, some proteins are relatively stable, and those are what we refer to as the cytoskeletal proteins. And so those are proteins that provide structural support to the interior and the exterior of the cell. So we're going to take a closer look at these proteins in terms of their function. What we see here in the green, some proteins act as receptors. So you see the triangle here represents a chemical messenger, and it binds on the outside of this particular protein, but the chemical messenger does not come into the cell. Its job is to bind to the protein, and that will initiate a signal to the interior of the cell. So two examples of chemical messengers include insulin and epinephrine. The second example here in the blue is that some of these proteins are enzymes. One example of an enzyme is the sodium-potassium ATPase. We know it's an enzyme because the suffix of this entire term is ASE. It's a protein that functions to accelerate or speed up a chemical reaction. And the one notable enzyme important for biosignal transmission is the sodium-potassium ATPase. Some proteins act as channels. One example of a channel is abbreviated as AQP, or an aquaporin. So some cells, particularly cells in the kidney, have what are called water channels. And so H2O molecules can move quite easily through these proteins that have been synthesized selectively to allow H2O molecules to move from one side of the cell to the other side of the cell. A second type of channel is called a gated channel, and these are not channels that are typically open, such as an alleyway between two buildings, but these are pathways that have a gate that open and close. Two examples of a gated channel includes the sodium channel and the potassium channel. Now, each of these ions have a particular protein specifically synthesized for that ion and only that ion. Other proteins serve as identity proteins. So one example are what are called MHC proteins, and cell identity proteins will recognize, for example, pathogens through certain structures on the outside of that pathogen, and will bind in an attempt to neutralize that. And these types of proteins in the membranes are found in the immune system. The last functional protein here are called cell adhesion molecules, or abbreviated as CAMs. And these are proteins, as you can see illustrated here on the far right-hand side, serve to connect cells to cell. One example of or one location of an example are cardiac cells. So cardiac cells that make up the heart do have these cell-to-cell connections, and that's an advantage in the heart because that facilitates the transfer of bioelectrical energy. Just the last few points here on the plasma membrane, just as a review. Once again, the plasma membrane is also described as a thin film of lipid and protein molecules. It's a dynamic fluid structure. And just one last, like as I indicated, one last review here. Here we see that the foundation of the plasma membrane is the phospholipid layer embedded with a range of proteins, and some of those proteins do have carbohydrate units attached to these proteins, and we would just simply call that protein a glycoprotein. So just to review, one final review here with respect to membrane proteins in terms of 4 basic functions. The first here is transport. 2 examples include glucose and amino acids. So these are considered to be very important nutritional molecules obtained in the diet that must be transported into cells for usage. As indicated, some of these proteins serve as enzymes, and the sodium-potassium ATPase is one such important enzyme. Transducing exterior chemical messengers to changing some function inside the cell is a signal transduction function, and attachment recognition. CAMs are an example of an attachment or recognition, or the immune system function is captured in this 4th basic function. One description of the membrane proteins are referred to as transmembrane. So when we talk about a protein in the membrane as being transmembrane, it has a component that spans the entire thickness of the membranes. And so the interior part of this protein that spans the membrane tends to be hydrophobic and are made up of nonpolar amino acids. And these tend to form generally what's called an alpha helix structure. It's in terms of protein structure, it's considered a secondary protein structure. And the alpha helix is ideal because it can interact favorably with the fatty acid tails of the phospholipid molecule. The hydrophilic or water-loving or polar amino acid components of those membrane proteins are typically found on the external or internal portion of the plasma membrane. Peripheral membrane proteins, not the integral membrane proteins, typically are located and connected on one side of the membrane or the other and do not interact with the hydrophobic core of membranes. And so these are proteins that are held to the membrane surface, usually through hydrogen or ionic bonds. And one such example on the inside of the cell is the cytoskeleton. So these are proteins that serve a structural or mechanical purpose of holding that very fluid cell together, and it allows the cell to bend and change shape up to a point. So it does provide the cell with flexibility, but structural strength as well. So this ends our first video on bioelectrical signaling.